All NEET aspirants, you are listening to podcast Botany for NEET by S. Ramakrishna. In this podcast, molecular basis of inheritance, the subtopic, the salient features of DNA. As the DNA is a double helical model according to Watson and Crick, as it is having two polynucleotide strands and sugar and phosphate which are alternately present and a nitrogen bases extended from backbone, that is sugar phosphate, more or less perpendicular towards the inside. And these two polynucleotide strands which are anti-parallel in opposite direction. In one strand 5,3, the other strand it is the 3,5. The 5 end it is having the phosphate moiety, third end it is the free 3OH sugar. Again in case of other strand show the same the polarity 3,5. So 5,3, 3,5 it is anti-parallel opposite. Next. In these two polynucleotide strands, the nitrogen base is paired through hydrogen bond. That is adenine, the purine pairs with thymine, pyrimidine. Guanine, purine pairs with pyrimidine, it is cytosine. Adenine with thymine 2 hydrogen bond, guanine with cytosine 3 hydrogen bond. So purine pairs with the pyrimidine, pyrimidine pairs with the purine, vice versa. That generates approximately uniform distance between the two polynucleotide strands because purine pairs with pyrimidine, pyrimidine pairs with the purine. Next, the two polynucleotide strands present in a right-handed fashion, a right-handed helix, a twisted ladder, and one turn of helix, that is the pitch of the one turn, it is 34 angstrom. So length of DNA is variable. Length of one turn, that is pitch of helix, is 34 angstrom. 10 angstrom is equal to 1 nanometer. So 34 angstrom is equal to 3.4 nanometer. 1 nanometer is equal to 10 to the power of minus 9 meters. So 0.34 into 10 to the power of minus 9 meters. That accommodate roughly 10 base pair. The distance between the base pair is 0.34. So approximately it is 0.34. So 10 into 0.34 comes around 3.4 nanometers. So the approximate distance between the two consecutive base pair is 0.34 nanometer. Next. So one turn of helix, that is one pitch of the helix, can accommodate roughly 10 base pair. Next. The stability of the DNA due to 1. The base pairs stack one above the other, that provides stability. The second, even though hydrogen bonds are weak, due to presence of many hydrogen bonds, it provides stability. It is the physical stability due to base pair stacking and the presence of hydrogen bond. And chemical stability because of thymine. So acquiring thymine in place of uracil by the DNA provides stability to the DNA. And fourth one, it is the deoxyribonucleic acid deficit of one oxygen at second carbon. It also provides stability to the DNA. In contrast to the RNA having oxygen at second carbon makes the RNA labile and highly degradable. Next, how can you differentiate the purine and pyrimidine? What is the reason for uniform approximate distance between two polynucleotides? Earlier we said approximate almost uniform distance between two polynucleotide strands because purine pairs with the pyrimidine, pyrimidine pairs with the purine, vice versa. Next. Purine dicyclic, pyrimidine monocyclic. Purine adenine guanine, pyrimidine thymine, cytosine. Next, the revolutionary aspect in case of DNA. Proposing the double helical model is one of the revolutionary aspects. The other revolutionary aspect is simplicity in explaining genetic implications. That is, two daughter DNA exactly similar to the parent DNA only after proposition of, that is, replication of DNA. Because according to Mendel, the progenies must be similar to parent. Here also two daughter DNA is exactly similar to the parent DNA. Why it exactly similar to the parent DNA? Because in parent DNA, the two strands act as template. Each strand synthesizes a complementary strand. So two daughter DNAs are found. One daughter DNA with one old strand, and that is parent strand, one new strand. And the mechanism, it is semi-conservative mechanism. Next, Francis Crick proposed central dogma of molecular biology. Dogma is any proposal or hypothesis is not questionable. That is what they call it is the dogma. So central dogma in molecular biology is DNA to mRNA transcription, mRNA to protein, it is translation, and this uniflow of information synthesizing the protein ultimately is heterocatalysis. So DNA through heterocatalysis regulate metabolical pathways by protein, expression of the character. And DNA to DNA autocatalysis maintain genetic integrity. So DNA through autocatalysis maintain genetic integrity, and through heterocatalysis it regulates the metabolical pathways by synthesizing the protein. What we call central dogma of molecular biology according to Francis Crick. There is a minor exception in case of reverse transcription virus. As in case of DNA replication, DNA dependent DNA polymerase that makes that DNA. But in this reverse transcription, RNA dependent DNA polymerase deoxynucleotide, the product it is cDNA, complementary DNA. That is called reverse transcription, also called teminism, after it discovered by Temin and Baltimore. And some numericals with related to this the DNA. We don't know the length of the DNA. Length of DNA is highly variable. Length of one spiral, the pitch of the helix is 34 angstrom. We know 34 angstrom can accommodate roughly 10 base pairs, that is 20 nucleotide, 10 purines, 10 pyrimidines. So we can say minimum number of hydrogen bond in one spiral 20, because all adenine thymines mean 10 into 2 20. Maximum number of hydrogen bond for one spiral as 20 nucleotide, 10 purine, 10 pyrimidine. If all guanine cytosine, 10 into 3, 30. So minimum 20 hydrogen bond, maximum 30 hydrogen bond. And number of carbon nitrogen rings in one spiral, 10 purines, 10 pyrimidine. Purines are dicyclic, 10 into 2, 20. Pyrimidines are monocyclic, 10 into 1, 10. So 20 plus 10, 30 carbon nitrogen rings for one spiral. Next, suppose there are 8 nitrogen bases in one strand 4, in other strand 4, it is a linear double-stranded DNA. In first strand 4 nitrogen bases, there are adjacent nitrogen bases are held through 3,5 phosphodiester linkages. So 3 phosphodiester bond. In other opposite strand, 3 phosphodiester bond. So 3 plus 3, 6 phosphodiester bond. So number of nitrogen bases 8. Number of phosphodiester bond 6. So it is n minus 2. The principle to calculate the number of phosphodiester bond when it is a linear double-stranded DNA, n minus 2. And to calculate number of phosphoester bond, each phosphodiester bond having 2 phosphoester. So 6 into 2, that is the 12. In first strand, it is having 1 phosphoester at 5 end, that is 1 phosphoester. On other strand, another phosphoester, we have to add the 2. So number of phosphoester bond in case of double-stranded DNA is n minus 2 into 2 plus 2. So 8 minus 2, 6, 6 into 2, 12, 12 plus 2, 14. So there are 14 phosphoester bond in a given data. Next, if it is circular DNA, the 4 strand, it is 8 in case of circular DNA. Suppose if you make the single strand into a one circle, there are 4 nitrogen bases means 4 phosphodiester bond. In this you make a one circle. 4 nitrogen bases, 4 phosphodiester bond, 4 plus 4 double stranded DNA, 8 phosphodiester bond in double stranded DNA. Number of phosphoester bond is equal to 8 into 2, that is 16 phosphoester bond in case of double stranded DNA. Next, suppose in a given DNA length there are 1300 hydrogen bonds. Length not given, 1300 hydrogen bond. In that 20% constitute adenine. Adenine number not given, 20% adenine. So you have to find out number of hydrogen bond, that is number of double hydrogen bond, number of triple hydrogen bond. In the data given 1300 hydrogen bond, you have to calculate how many double hydrogen bond, triple hydrogen bond, length of the DNA, number of phosphodiester bond, number of phosphate, number of carbon nitrogen rings. So you sort out the problem and mention in case of the comment box.